Welcome to a lecture on cell division, and we'll talk a little bit about cancer and its association with cell division. First off, today we're going to focus on mitosis, which is a type of cell division in somatic cells. Somatic cells are all the cells in the body except for those associated with the germline cells, or sex cells. Okay, so all cells except for your sperm and your egg are somatic cells, and they are diploid, meaning they have two sets of chromosomes. Right, one from the mom and one from the dad. They have a paternal and a maternal set of chromosomes. We call this 2N. In cell division, you have a cell that divides and produces clones. Okay, we call it parent to daughter cells. So a parent goes and becomes two daughters. The daughters are clones, they're exacts, and they go from 2N to 2N, right? Diploid to diploid. They're exact copies. So mitosis, spell it for you up here even though it's on the next page, okay, or mitotic cell division, is division of somatic cells. It creates clones, and they are diploid, meaning they have two sets of chromosomes. Now, what's the purpose of cell division like this? For repair, for growth. Right after an egg gets fertilized, it goes through massive cell division for a long time to produce a baby that is born during labor. And we have cell division still going on in our body today from our stem cells in each individual tissue. Where we have stem cells, we have repair constantly in a lot of our tissues. The only area that mitosis isn't heavily happening is in our nervous system with our neurons, because it's while it does happen in the peripheral nervous system, central nervous system is still we're still trying to study that one, because we don't tend to have any continued mitosis after a certain age in our central nervous system, which is why we lose function when we have a damage to our spinal cord. Now, in order to understand cell division, we have to look at the cell life cycles. Yes, cells have life cycles, and they're controlled by the body. So let's look at the basic cell life cycle here. And I'm going to use this for a different stage. Okay, so if this is our cell life cycle, there's a couple stages we're going to look at. This stage, I'm going to use red for this. So this stage here to here, okay, and all the way around is interphase. So red is interphase. And that's 90% of the cell's life is spent in interphase. Now there's actually three parts to interphase. There's G1, S, and G2. G stands for gap 1 and gap 2. In gap 1 and gap 2, they're kind of just going about their normal functions, everyday cell life. S is specific to DNA duplication, right? Because if we're going to divide a cell at some point, we have to duplicate the DNA so we can create two exact copies. The blue up here is our mitotic portion of the cell life. This is where cells are actually going to go through cell division. Now, how do we control this? Well, there's things that actually control the cell. We have, let me move down here a little bit so I can write. We have proto-oncogenes. And these are genes that produce molecules that control normal cell division. Okay, so these are genes that produce molecules that control cell division. So they would produce proteins maybe that would cause cell division to occur or inhibit cell division. Okay, and we have different factors. We call them growth factors and growth inhibitors. Okay. These are molecules that control cell division, and they can be produced by proto-oncogenes. Now, another type of gene, and I can't go down any further, so I'm going to just erase this. Well, I don't want to clear ink. Let's just go back here. Actually, let's just go back. Sorry. Slowly deleting all of this. So those are the proto-oncogenes. We also have tumor suppressor genes. Which are genes that produce molecules that put on the brakes, so they're the cell division brakes. So they stop. They're specifically for stopping cell division to prevent tumors or to prevent the growth of a tumor. Okay, so they're tumor suppressor genes. Those genes help code for the growth factors or growth inhibitors and other molecules that help control cell division. They help control the cell life cycle. All these phases, G1, S, and G2, are controlled by growth factors or a growth inhibitor that may stop the cell from continuing into mitosis, the mitotic cell division portion. Now, sometimes, which we'll talk about in a minute, the proto-oncogenes can become mutated into oncogenes, which is what causes an acceleration of cell division, causes basically these signals in the cell membrane to initiate cell division constantly. Because what happens is molecules will bind to a membrane, and that sends a secondary messenger cascade through the cell. That's a message that gets sent through the cell to tell the cell to begin cell division. If this were to get stuck on, which is what happens with oncogenes, then it will continually divide, and that's how you can get cancer. Alright, let's look specifically at mitosis and cell division. There's four main steps to mitosis. There's prophase, metaphase, anaphase, and telophase. Easy acronym is PMAT. Okay, so this is prophase, metaphase, anaphase, and telophase. And there's something actually occurring here that we'll talk about here in a minute. The first step, prophase, what happens is you lose your nucleus. So you notice there's no more nucleus present. You can't really see a nucleus that's broken down. I'll show make it look like it's broken down. Your chromosomes also pair up in what we call sister chromatids. Okay, and I'll show you an example right here. For example, if this was mom's number 1 chromosome, or maternal 1, this is maternal 1 copy. So this is maternal 1 copy. These are sister chromatids, we call them. Okay, they're paired up, the original and its copy. So when we duplicate the DNA, each chromosome lines up with its copy. We call those sister chromatids. Okay, and that is spelled like this: sister chromatids. That's it. TIDS at the end. And then we also have these right here. The yellow is the movement of the centrioles to the end of the cell and sending out microtubules. As we get into metaphase, all the sister chromatids line up across the equator, or the metaphasal plate, and the microtubules from the centrioles come out and attach to each side of the sister chromatids. And that's metaphase. And then when we get to anaphase, these sister chromatids get separated, going their opposite ways. So the original goes one way and the copy goes the other, so that we can have two duplicated set of DNA in each cell. Right, we want them to be clones, we want them to be perfect. And then as we get to telophase, the DNA is now on its own side. It will start to reform the nucleus. The microtubules will disappear. The DNA will actually uncoil into what we call chromatin form. It just looks like spaghetti noodles in the nucleus. It's not condensed anymore. And then we get in here, in an animal cell, okay, we get a cleavage furrow. I'll spell that for you. Cleavage furrow. Alright, we get a cleavage furrow in there, and that helps pinch the cell completely apart, the cytoplasm apart, and we get two diploid daughter cells, and that is mitosis, the simple form of mitosis. Now, when we're looking at this cell division and cancer, what happens is, and we'll get more into this later in class when we deal with some case studies, but in cancers we get these mutated DNA. During DNA duplication, the genes mutate, and we get these oncogenes that basically put a on switch on. They hold the pedal down, and this causes rapid abnormal cell division. And I'll show you a video in class, but it causes the cells to grow at an abnormally increased rate, much faster than normal cell division. I believe if we're looking at it, when we look at the video, I believe on day 2 when there's like 8 cells from normal cell division, there's almost 100 already from cancer cells. So cancer cells grow rapidly. When you get a tumor that's growing, you can have a benign tumor or you can have a metastasizing tumor. Okay, and that metastasis is really what is significant of cancer. Okay, and I'll write that out for you here. Let me clear the ink on this page. So you get benign, okay, or you have cancerous, and the cancerous, key with the cancerous is it metastasizes. Metastasis. Except I spelled that wrong. There should be an A. Metastasis. And what that means is the cancer cells break off. The cells break off and spread through the body. Okay, and that's cancerous, right? Cancer can spread. So what happens is these tumors grow, the cells break off, they spread through the body and infect other tissues. As tumors grow, they take over these tissues and the other cells can't function, they die off, because what happens is these cancerous tumors, these cells, actually cause angiogenesis. What that is let's say I have this mass of cells here. Well, these cells need nutrients to continue to grow and grow rapidly and take over their areas. So what they do is they cause angiogenesis, which is the production of blood vessels to their areas. So they'll actually cause blood vessels to come in to their areas and bring nutrients. So these nutrients will come in. So these black things are our nutrients. They go into the cells, and the blood continues to flow out, and nutrients keep flowing in, and it's a new tissue in the body, and these are new blood vessels that are feeding the cancer. They are finding that, you know, this cancer can just take over our body and regulate its own function, and it allows it to take over certain tissues in our body. And they're finding more and more about different parts of our cells that contribute to this. In fact, I was just doing some research on a Tay-Sachs disease, and there's a carb and fatty acid molecule that actually helps in allowing different cancer cells to communicate with each other from tumors, allowing them to communicate with molecules. So it's interesting research they're still finding. Okay, but make sure you know the connection between the cell division and the cancer. Okay, understand the growth factors, growth inhibitors, how we're supposed to control the cell cycle. When we get these mutated genes, we get these oncogenes, we can't control the cell cycle. The gas pedal gets put down. We get cancer cells or tumor cells growing. If we get metastasis, we get cancerous cells, and then they produce blood vessels, which is, well, they signal the production of blood vessels, which is angiogenesis. So make sure you identify those key concepts. In class, we will be looking at a case study to cover this material as well. And then we will, in the following week, we're going to go into meiosis, which is another type of cell division, which is what we use to produce sperm and egg.